Welcome those of you in the room here at CBL. Welcome also those who are joining us on Zoom. My name is Tom Miller. I have the honor and privilege of being the 6th director of the Chesapeake Biological Lab. I have thoroughly missed these Science for Citizens lectures in person, and I can't tell you what a delight it is to be back sharing our science with all of you, talking about what we do and why it is important. This is the 3rd of 5 presentations that we have done since the end of September. We talked about an organizing principle when we put this seminar sequence together, and we decided we were going to call it our greatest hits. And so we talked about crabs in the first week. We talked about sturgeon last week. As you'll see from the screen, we're going to talk about terrapins tonight. Next week, next Tuesday, a talk called Long Time No See. We'll talk about bottlenose dolphins in Chesapeake Bay. And then in the final presentation, one of our long-term faculty members, Dr. Walter Boynton, will come and talk about the changes he has seen in the Chesapeake Bay in his 50-year career and the changes that he thinks still need to occur. For those of you in the room, I have to thank Toyota Southern Maryland and Team Hyundai for the tea, coffee, biscuits. For those of you on Zoom, I assume you can get tea, coffee, biscuits of your own. Tonight, it is my real pleasure to introduce a colleague of mine, Dr. Chris Rowe. At heart, Chris is a naturalist. Nothing makes him happier than being outside communing with nature. And he is one of the people who, as a graduate student, or he's representative of a class of people who, as a graduate student, I was always insanely jealous. They knew their natural history, they knew their communities, they knew the animals that they were working on in a very deep and profound way. Chris has spent his time to deepen that knowledge throughout his career working on reptiles and amphibians. He works on frogs, he's worked on snakes, he's done a little bit on shrimp. It's deviated ever so slightly once. But it really is amphibians and reptiles that are his passion. And he's going to talk tonight about some peculiarities of the biology of those two groups of animals that makes them potentially very susceptible to climate change. I'll remind everyone on Zoom that this is being run as a Zoom conference. So unfortunately, at the end of the presentation, you will not be able to unmute and ask questions, but you will be able to type your questions in the chat box. And at the end, I will take questions from the room and alternate those with questions on the chat box. So if you have questions during the presentation, don't hesitate to type them in, and we will address them all at the end. And so without any further ado, it is my pleasure to introduce Dr. Chris Rowe. Chris.
Hi, everyone. Thanks for having me. I'm sure I'm deserving of that introduction, but let's see if I can.
You're good to go.
Okay, great. Is this a touch screen or is this?
No, the mouse, just drag it. So there you go.
Okay, trying to get that out of the way. All right. First of all, I'd like to thank Sarah Brzezinski for coming up with a catchy title for this talk. I don't have that great of a sense of humor, so she's better at wordsmith than I am. So I'd like to talk to you tonight about the Diamondback terrapin, a turtle, a reptile that many of us are familiar with, at least at some level. But before I talk about the Terrapins specifically, and some of the things that we've been doing with our research involving it, I want to talk a little bit about the environmental context in which our studies are taking place and the environmental situation that's causing us to look into certain issues with this animal. So
You move the mouse to just hovering on the terrapin, and now click it up and down, it should work.
there we go. Great. So the situation today, it's getting warmer. I think we all hopefully by now recognize and accept this as a fact. And this is an illustration of the global average temperatures of, Maryland's average temperatures of the past roughly 100 years. And what we see is essentially it's been increasing from left to right. And if we look, if we look at our, I came in, okay, so. All right, almost, I'm not getting anywhere here. Previous. You know, I've been teaching on Zoom for about 3 years now. I think that at some point I learned all the 2 months.
Which one we got?
Yeah. Oh, well, awesome. Thank you. Okay.
Could you adjust the volume up as well, please?
How about if I pull this over closer to me? Is that any better? All right. Cool.
Okay.
So yeah, it's getting warmer. Okay, and that's one of the things that is driving some of our work, because Diamondback terrapins respond to temperature in ways that some other animals don't. Diamondback terrapins, like all reptiles, like actually about 99% of animal species, if we include the invertebrates, are ectothermic, they're cold-blooded. So changing the temperature can mean a lot to them physiologically. Okay, unlike us, we of course maintain our temperature within a very tight bound. And whereas the terrapins and the other ectotherms, their body physiology changes as the environmental temperature changes as well. So we have a situation where it's getting warmer, and associated with the warming is the fact that sea levels rising. On the left, I have a figure showing the global sea levels for the past 1,000 years. And it's pretty clear that as we approach today, we see a really dramatic increase in sea levels. For some species, especially animals like Diamondback terrapins that require shallow nearshore habitats, sea level rise can be a big deal. And so that's something else I'll be talking about today as well. On the right, I'm showing the sea level trends in Chesapeake Bay. And what we'll see is that since this is normalized in 1980, so the sea level is set at 0 in 1980. And so since 1980, in Baltimore, the sea level is up roughly about 9 inches. And that's in the past 40 or 50 years. Which is not surprising given that first graph showing how quickly temperatures rising across the globe. The third thing I want to establish before we move on, and this is also in one way or another associated with the change in climate as well, is that wildlife populations are declining. Some of you may have read about this. It's in popular press. There's been a lot of discussion about this, especially over the last 4 or 5 years after a few studies came out, got a lot of attention. But on the left is a panel showing The percentage of species that have been documented to be declining in their population sizes, vertebrates only, this is looking at across the different parts of the world and the world as well. You see in the tropics, especially in the neotropics, population declines have been disastrous. So it's getting warmer and sea levels coming up and wildlife are disappearing. That's not a situation that I think we ever wanted to be in. On the right, since we're talking about terrapins today, I thought I'd focus in on reptiles a little bit. On the right is a figure showing the population status of European reptiles. And the point of that graph is essentially that a little under half of the reptile populations in Europe are documented to be declining. Europe isn't special compared to the rest of the world. This is happening globally. In fact, if we look at reptiles specifically around the world, about half of the reptiles are currently considered to be threatened or vulnerable. Threatened means that they have the potential to become endangered and ultimately extinct within the near future. What I'm showing on the left is the categorization of different reptile groups. As you see, the number of species is listed here. This is presented by essentially by the orders of the reptiles. There are 4 modern orders of reptiles living today. 5 groups there because one of the orders is split into 2. That's the snakes and the lizards. But there are a few groups, the snakes, the lizards, and the turtles, that are considerably more diverse than the others. However, those groups that are the most diverse are also the ones that we know the least about. Okay, so over 3/4 of the reptile species in the world, we have no information as to whether their populations are increasing, decreasing, or remaining stable. Why are reptile numbers declining? Well, there are a lot of reasons, actually. This isn't an exhaustive list, although I think I've hit most of the major issues here. But up on top of the list is probably the most important human activity that's influencing not just reptiles, but wildlife in general globally. And that's loss of habitat. Okay. Habitats are being destroyed for human purposes. They're being fragmented and broken into smaller pieces, which makes it difficult for animals to access them the way they should. And the habitats are being generally altered in one way or another that makes them unusable or at least less usable by wildlife. See here, for example, of course, deforestation. Deforestation is a really obvious source of habitat alteration that can clearly affect wildlife, but other things that we do, especially things that we see locally, for example, changing shorelines in order to protect our properties, is isolating nesting habitats from terrapins and turtles, and also influence whatever other communities would typically exist along a natural shoreline. Pollution, of course, is a problem globally. We are in a situation today where there are no unpolluted habitats on Earth. You can find man-made chemicals everywhere you look, if you look hard. From the Arctic, from the North Pole to the South Pole, from the deep ocean to the surface ocean, to the terrestrial environments, the air around us, there's pollution everywhere. Disease is an issue for the reptiles. It's an issue for all wildlife, but reptiles, amphibians as well, but I'm focusing on reptiles today. But there are certain diseases in certain populations that are really important to these species that are there. Invasive species. We as humans, we like to move things around. Don't necessarily always put them in the right place. And when we move species from one habitat to another, we put them in conflict with natural species that are there. can be altering the competitive relationships among individuals, can alter predator-prey relationships, or we can introduce species that may be very unlike our species of concern. For example, plants. But we introduce species that can alter, that change the habitat physically. Like I've shown here, this is Phragmites highlighted right now, and it's a plant that's invasive and it tends to change the shorelines, and therefore it makes them different from what they were, and therefore not usable by the same species that naturally would use them. Harvest. So believe it or not, reptiles are harvested intentionally for food in many areas. Many of you may have eaten turtle in the past. Snapping turtles is popular. Diamondback terrapin was popular once. We'll talk about that in a bit. But reptiles are also used for other things besides just food. Homeopathic medicines, you look in Asian cultures, there's a lot of reliance on reptiles for some of their homeopathy and some of their traditional medicines. The exotic pet trade removes a tremendous number of wild animals from nature every year. If you are interested in exotic pets, please make sure that they are captively bred. Because for about every 10 animals that are captured in the wild in order to put them into the pet industry, about 9 of them don't make it that far. Okay, so harvesting animals for the pet industry is a big problem worldwide. Bycatch. That's an unintentional means of harvesting animals. Bycatch means harvesting an animal that you didn't target. Okay, for diamondback terrapins, a big issue is bycatching crab pots. And we're going to talk about a little bit about that today as well. Roads and boat traffic, we're all familiar with roadkill. Boats in shallow waters can affect animals like terrapins. One of the things that is kind of unique about reptiles relative to most other groups of animals, but perhaps the exception of the arachnids, spiders and their kin, is that reptiles are basically persecuted by humans. Okay, Homo sapiens do not like reptiles. Okay, we treat them badly, we kill them for fun, we kill them simply to get them out of our way. Their reasons for this go anywhere from simply fear, superstition, to you can come up with some of your own ideas. Finally, what I end with here is climate change. So climate change is represents several really big processes, things that are operating in large spatial scales, and they're interacting with many of these other issues to make the reptile populations somewhat fragmented relative to what they used to be. So let's talk about the terrapin specifically. So here in Maryland, the diamondback terrapin extends from the Gulf of Mexico to New England, but there are 7 subspecies across that region. Here we have what's called the northern diamondback terrapin. Okay, and its range runs from the Carolinas up to New England. Terrapins are the only estuarine endemic turtle that we have in North America. When I say estuarine endemic, I mean it's an animal that lives only in the estuary. Okay, the estuary being like the bay, brackish water. Okay, it's saltier than fresh water, not as salty as seawater. That's a challenging place for animals to live because things like salinity, dissolved oxygen, things like that fluctuate a lot in those systems. So it's a difficult place to live. The terrapins have specialized in it. In fact, only 3 turtles in the world can do that. The terrapin is the only one in North America. The terrapin's closest relatives are turtles that you probably have seen or are familiar with. They're a member of this family called Amididae, which is the family of pond turtles or water turtles. If you go to a pond or a lake in the summer and you see a bunch of turtles on a log and they see you, they scurry into the water, those are probably relatives of terrapin. Okay. Terrapin, so originally, from an evolutionary perspective, the terrapin was a freshwater turtle, but it specialized in order to live in the estuary. One of the specializations is that it's come up with a way of getting rid of extra salt from its body. Okay, and that's through a salt gland near the eye, called the lacrimal salt gland. In any case, Diamondback terrapins reproduce in the summer, and the females typically come up the beach 1 to 3 times per summer to lay a nest. And many other turtles only nest once per year. Diamondback terrapins do it multiple times. They typically lay smaller nests than some other turtles. So on average, roughly 6 to 20 eggs are laid in a nest. Around here, the average value is around 12 to 13 eggs per nest. So if they lay a nest twice a year, they can get up to maybe close to 30 eggs in the summer. They're long-lived. Turtles are famous for living for a long time. Terrapins are thought to live for perhaps 40 years. I would not be at all surprised if there are older individuals out there. I personally know of at least one individual that's close to 20, or actually well over 20. There's an animal that I captured in a field site in 2004 who was a mature adult nesting female, and I caught her again last year. And it takes these animals 6 to 12 years on average to mature females. So you do the math, it was getting pretty old. Okay. So they live a long time, and it takes them a long time to mature. As adults, there's a sexual dimorphism among the sexes. Sexual dimorphism means that the two sexes look different. Okay. And the way the terrapins look different, well, there are several ways that they appear different, but the major way is in their size. Females are about twice the size of males. I've shown here on the right a female beside my lab notebook, or my field notebook, so you can get an idea of the size. So the females, a really large female is around 9 inches or so. Okay, males are down around 5 to 6 inches. Their ecology in terms of feeding is they tend to feed mostly on invertebrate prey. Mollusks seem to be preferred generally. And when available, the marsh snails, particularly the marsh periwinkle, seems to be a preferred food source. At the bottom of this slide, I've shown a picture of some of these marsh periwinkles. Basically, what these animals do is they climb up and down on the stems of the salt marsh grasses, and they scrape off the stuff that grows on the surface. Okay, so the microbes, stuff called paraphyton, they scrape that off, and that's what feeds them. The terrapins come by, pop their heads up out of the water, and pluck them off of the grass and have a meal. There was a day when the terrapins were more abundant that you could go to a marsh and you could hear popping, and that was the popping of the shells as the terrapins would crush it with the snails as they came through the fed. You don't hear that much. One interesting fact regarding that feeding strategy is that in the process of these snails feeding on the marsh grasses, they actually damage the grasses. So the snails don't eat the grass, but by feeding on the surface of the grass, the way they feed is they have this really rigid mouth structure called a rhangulites. It's made of silica, and it's used to scrape the stuff off the surface. But what it does is it oftentimes also removes some of the plant material as well. So the grazing by the snails actually can cause the marsh grasses to die or to not grow as well as they should. And so there's been some work in New England that suggests that Diamondback terrapins are actually facilitators of marsh growth because of their activity of removing these snails, or at least cropping their populations back to the point where a healthy marsh can persist. Predation is a big deal for diamondback terrapins, especially when they're young. Okay. These are, so these females come up across the beach and they nest on high, in sandy areas above high tide. And usually, in many areas, 9 out of 10 nests are destroyed by predators within the first day or two after growing. From an ecological standpoint, they're an incredibly important food source for other wildlife. But when we're looking at it from the terrapin's perspective, it's a real bummer, right? So you got this huge amount of predation occurring on the eggs. If the eggs are lucky and the fox or the raccoon or whatever doesn't smell the nest in the first few days, well, the nest may be safe. But then comes another important transition in life, and that is hatching and crawling away. And baby terrapins are about the size of a quarter. Okay, so there's this little quarter-sized chunk of energy walking around that everything out there that's bigger than it wants to eat. Okay, so predation rates are extremely high on the hatchlings as well. It's very hard to estimate the predation rates at this age because it's hard to track individuals. But there's some work that suggests that probably less than 8% of the animals survive the first year. Okay. So again, from the perspective of the terrapin, that stinks. But from the perspective of a raccoon or a fox, that's great, right? So and that's ecology. That's how things fit together. We're looking at it from the terrapin's perspective today. Across the range, the diamondback terrapins listed as vulnerable. Again, I mentioned vulnerable and threatened, these categories that could suggest that the animal populations are not in as good a shape as they could be. And most of the reason for the diamondback terrapins declines in many areas is because of both intentional harvest through commercial fishing and bycatch and crab pots. Now, those are two major sources of mortality for the adults. Fortunately, as we'll see, commercial harvest of terrapins doesn't occur, at least not in Maryland. However, crab pots are still used frequently in Maryland, and they still catch diamondback terrapins. Finally, I probably don't have to tell you this, but the diamondback terrapin is the Maryland state reptile, and of course, the mascot of the University of Maryland College Park. So let's talk about terrapins and us, terrapins and Homo sapiens. I've alluded to this already. Terrapins are harvested, both intentionally, or they have been harvested both intentionally and unintentionally throughout their history of interacting with humans. And the reason is that terrapins taste good. People like to eat terrapins. Terrapin soup was a delicacy for the times during the periods that it was extremely, when they were abundant and when they were available for food. Native Americans relied on diamondback terrapins as a highly available source of food. Then when the European colonists came, they made use of the terrapins as well, again, because of their abundance. They were almost considered like a trash species. There were so many of them. They fed them to the Continental Army. They fed them to slaves. But lo and behold, as time went on, terrapin meat became quite popular both here and in Europe. Which means once a wild species becomes popular as food for humans, typically that means its populations are not going to be what they could be, and that's happened across the board with the terrapins. I have a few statistics here from catch records. While diamondback terrapins were harvested commercially in Maryland, and what we can see from the catch records is that there were periods of high abundance, high capture, followed by long periods of low capture. Sometimes low capture means that the populations are small. Sometimes it means that they're just not out there looking. But for one reason or another, there's been periods of very high harvest pressure on the terrapins, which ultimately resulted in periods when there were many fewer to catch. Basically, since the 1950s through until harvest was outlawed in Maryland, the harvests were down to a fraction of what they had been at their peak in the past. So here's the other issue with harvest, and that's this unintentional harvest in crab pots. Diamondback terror. So if you're, I assume most of you are familiar with crab pots in here, but perhaps maybe our cyber audience isn't. Crab pots are basically rectangular or square cages, and they have funnels that the crabs crawl in to get the bait. They're set underwater. The problem comes for the terrapins when the terrapins smell the bait as well, because the terrapins crawl in the crab pots as well. Well, unlike crabs, terrapins breathe air. And so when terrapins get into a crab pot, and that crab pot isn't like immediately pulled from the water, the animal has a very high chance of drowning. And so there's active crab pots themselves catch a lot of crabs or a lot of terrapins. But one of the big issues that we see here in the bay and in many other areas where the blue crab fishery is important is that oftentimes crab pots are lost. Okay, so when a fisherman puts out his crab pots, he puts a float. He has a rope with a float, right? And so he can go out in his boat and find his float and completely engage against crabs. Well, frequently that float gets lost. Somebody comes by in their speedboat and cuts the rope or something like that. And that pot is left sitting there. And it's not tended anymore. And so what the pot does is it sits there and it gathers terrapins and it kills them. And that's what some of these pictures are showing here is bycatch mortality in what are called ghost pots, which are pots that have been lost. There's sometimes dozens and dozens of animals will be killed in one of these lost or ghost crab pots. So given that there's been harvest, either intentional or unintentional harvest for so many years, and because there have been periods of abundance followed by periods of low abundance, it was only natural that there would be some sort of regulation placed on the fishery itself. One of the regulations, and there's something that's still on the books actually, is one that causes me to pause. And that is that legally, if you're a lawyer, terrapins are fish. Okay. According to the state, they define fish basically as any aquatic, whether or not it's really a fish or not. Problem is, that means it's managed as a fish, but terrapins are not fish. I think those of you who fish for sport know that the regulations typically require that whatever species you're catching has to be a certain length before you can keep it, right? So there's a minimum length, and if it's smaller than that, you have to throw it out. If it's bigger than that, you can keep it. Well, given that terrapins are fish, that's how they were regulated as well. So in the early 20th century, the state set a minimum size for harvest of 5 inches, and in the 70s, they increased that to 6 inches. Now, here's the issue. Because terrapins are not fish, they don't necessarily fit into the same regulatory strategies as fish as well. Remember that sexual dimorphism I told you about. where the females are twice the size of males. What happens when you tell your fishermen you can only keep the big ones? They can only keep the females, right? So basically, throughout the entire 20th century, the commercial harvest was removing the females from the population. And anybody out there knows that population takes both males and females, and when you start taking out the females, especially the large reproductive individuals, the sustaining the population is going to be an issue. Fortunately, in the early 2000s, actually shortly before the commercial terrapin industry was shut down, they changed the size requirements somewhat in order to avoid catching some of the largest animals. So in the year 2000, there was an effort made to help alleviate some of the issues with accidental catch, with bycatch and crab pots. And so in year 2000, the state of Maryland put out a regulation requiring that recreational crabbers who put crab pots out have to put what are called BRDs or PEDs. These are devices to keep terrapins out of the traps. Okay, BRD means bycatch reduction device, PED means turtle exclusion device, whatever you call it. Basically, it's a rectangular piece of plastic or steel that sits over the mouth of the trap, allows the crabs to slide through and get captured, but it's too narrow for an adult turtle to get through. Okay. In the state of Maryland, if you own waterfront property, you are allowed to put 2 crab pots out off of your shoreline. Okay. Those are the crab pots that are regulated. Those are the people that have to put these TEDs or BRDs on their pots. Sadly, according to DNR, somewhere around 50% of the recreational crab pots out there don't put BRDs on. These things cost about $1. Something else you'll probably find interesting is that commercial crabbers are exempt. So the thousands of crab pots that go out there to the commercial industry every year don't have these excluders on. Right? I'm not going to get cynical right now and tell you why I think that's true. But let's just say it's a fact that we're trying to live with. Finally, in 2007, commercial harvest was outlawed in Maryland. So theoretically, a lot of the pressures on terrapins may be at least somewhat alleviated. We're not harvesting them anymore intentionally. We're not going out and bringing in thousands of animals to make soup. And some very small fraction of crabbers are avoiding catching them because of the TEDs. So whatever little progress that is, it is progress. The problem is there are a lot of other things going on right now that are, say, throwing gasoline on the fire. And the biggest issue right now are the responses that the animals are having to change to the increasing temperatures as well as the rising sea levels. So what we do when we look at the Diamondback terrapins is we take 2 angles, 2 approaches to it. We look at the physiology of the animal, we look at how they function, how they work. And we look at their ecology, how they interact with other species or other individuals of their own species. And what we do is we frame our questions to ask, how does climate change influence these features of the animals? And is it significant enough that we should have concern? So let's start with physiology. As Tom promised, we're going to talk about sex a little bit. But this is a rather unusual sexual strategy that occurs in terrapins. Terrapins, as well as most of the other turtles, all of the crocodilians, and a few, other species here and there, possess what's called temperature-dependent sex determination. What that means is that when a female lays her eggs, she digs her nest and lays her eggs. Those eggs are neither males nor females. Okay, so it's not like us. Us, you know, we have the X chromosome and the Y chromosome. If you get two X's, you're a biological female. If you're an X and a Y, you're a biological male. There's no X's and no Y's in the terrapins. Okay, they're neutral. So what makes them turn out to be a male or a female? Well, the answer to that is the temperature of the nest during a critical period of development. What happens is that this is a fairly complex phenomenon, and there's a lot of work going on into the nitty-gritty of it. But what appears to be the case is that the temperature somehow influences the activity of a very important enzyme called aromatase, which is an enzyme that causes male hormones to be oxidized in female hormones. Okay, so when aromatase is around, and there's a lot of aromatase, the male hormones become female hormones. The point is, aromatase is high, we produce females. And temperature seems to be either affecting the transcription of that from the DNA or the activity of the compound itself. So we've got an animal here that has no genetic basis for sex, but the sex of the offspring depends on the temperature of the nest. I think you can kind of put two and two together and figure out where we're going here, right? Because slide number one that I showed you today was that it's getting warmer. And it's getting warmer, the nests are getting warmer as well. The Diamondback terrapins display a fairly common pattern for animals that have temperature-dependent sex determination. And that's a pattern in which cool temperatures produce males and warm temperatures produce females. This graph is showing that there is a temperature here called the pivotal temperature, at which half of the eggs in the clutch will turn out to be males and half will turn out to be females. So that's an important temperature for us to know if we're trying to predict how many males and females will be in there in the future. And then as you diverge on either side of that temperature, as you get cooler, you go more and more males and warmer, more and more females. And at some temperature, you produce only males or only females. Okay, so one of the real drivers of our work now is trying to figure out what are those temperatures. Okay, at what temperature do we get half males and half females? At what temperature do we produce only females or only males? Now, the temperatures shown on this graph are actually published values for Diamondback terrapins. However, our work suggests that those values don't apply to our population at least. And so we're kind of back at square one where we're still just defining the basic temperature versus sex ratio relationships in these animals. How are we doing that? Well, we've been working since 2018 across the river at the Hudson Naval Air Station in Lexington Park, where there's a really nice breeding nesting area and a marsh area where the terrapins live and where the terrapins nest. And what we've been doing is we've been placing data loggers in the nests of the terrapins so that we can measure every 15 minutes from the time the eggs are laid to the time the eggs hatch, what's the temperature of the nest. Because until now, we didn't even know what the temperature of the nests were. Well, as you can see, the temperatures in the nests follow a complex pattern. They go up and down every day, just like air temperature does, only not as severely as air temperature changes. What I'm showing here on this graph are the temperature traces from 4 of our nests from last year. And basically, and the point is essentially to show you that the temperature in the nest is not stable. temperature in the nest varies. It varies from day to day, and it varies within a day. And trying to figure out what part of that distribution is the important part for sex determination has been a real challenge and something we continue to work on. Across the river at our field site, we also work with folks at the from the Navy Natural Resources Group who've been running a terrapin conservation program where they've been protecting nests from predators over there. As I mentioned, predators and nest predation is a really big problem. Since about 2016 or so, a group of volunteers over there has been putting cages around the nests, over 100 nests a year, in order to keep predators out. So that's a good thing. We're seeing a lot more input of hatchlings in the population than we would otherwise. Working with these folks has been, of course, very beneficial to our work as well, because we're all working on the same population, sharing information and what have you. So I mentioned that there's a brief period of time during development that the temperature influences sex. It's roughly between 40 and 50% of the way through the incubation period. And the period is what we call the TSP, or the thermally sensitive period. And I have that highlighted here. And what you see, so this is a temperature trace from one of our nests. And as you can see, as I mentioned, it goes up and down on a daily fashion. The long red dotted line shows the seasonal or the incubation period mean. So that's the average temperature of that nest. But that average temperature doesn't really mean anything in terms of the sex determination. The important part is the temperature during this narrow period of time, this TSP. Okay, and in this case, the TSP mean value is 21.7, whereas the overall mean was considerably less than. So what we've been able to do is we've been able to record the nest temperatures and calculate when in development approximately temperature is important to sex determination, and from there come up with, well, what was the temperature of the nest during that time? And that's what I have plotted here. Being a scientist, I love graphs, so I have to apologize for this because it's going to get a little hairier on the next slide. Basically, these are the average nest temperatures during that narrow period during which sexual determination occurs. And this is 33 nests collected or monitored over the past several years. And what you can see is that, well, what I have marked in the heavy dotted line is the purported or the literature value for the 50-50 sex ratio, okay, 29.3. Assuming that that value is right, we know it's not, but let's assume it is. If that value is right, we can look at, well, how often was the mean values above that, and how often was it below that? When it's above that, we're producing most of the females. When it's below it, we're producing most of the males. And if it's outside of that narrow range at which both males and females produce, then we see production of only one sex of the other. So looking at this graph, what we see is that, again, assuming that this pivotal temperature is accurate, in this group of nests, 61% of those nests were female biased, would have produced more females than males. And 58% of them would have produced only females, okay, over the past 5 years. Well, what we're interested in, though, is this change, what's happening as the environment changes. So what we're interested in is what's happening in the future. And so what my next graph is meant to show is how many males or what proportion of males and females would be produced if temperature was a little bit warmer, let's say 2 degrees Celsius. And that's the yellow bars. So what we see is just a very small change in temperature, adding 2 degrees to that nest temperature during that critical period of time now produces 91% female bias nests. Of those 82% produce only females. What's the big deal about having too many females? Well, in many populations, it's good to have a lot of females around because oftentimes males can make more than one female. But the issue is, at some point, if we continue to make females and not males, eventually we're going to run out. We're not going to have enough males. Now, that's a question that we don't know the answer to, is how many males is enough? We don't even know how many females a male needs. Okay, problem could be 3, could be 10. And the other thing is the females are able to store the sperm, sometimes for a year. So maybe she only runs across a male occasionally, but that's enough. Okay, so there's a lot of complications in figuring out what's going on here. But those are the things that we have to account for as we start doing the math, what have you. So what we're doing now is we're also now, because that the temperature value, that 29.3 degrees, the pivotal temperature, I said, doesn't appear to apply to our population. We're doing a series of incubation studies right now in order to determine for our population exactly what temperature it takes to make half males and half females. To make all males, we're to make all females. And what we're doing is, because we're measuring these nests in nature, we can use the variation in the temperatures that we see in the nests in our incubators as well. So we can mimic the natural situation in the laboratory. And hopefully, what we'll be able to do is track down the actual values and move forward from there. So in a nutshell, terrapins produce males at low temperatures and females at high temperatures. We also know that it's getting warmer. And so it doesn't take rocket scientists to realize that in a warmer future, we're going to have more females than we have today. Again, how many is too many? How many is not enough? Into the ecology. So now we're looking at the animals from the perspective of their environment. And what we're focused on here is how their critical habitats change. Okay, so I mentioned that the terrapins like to nest on these sandy areas above the beach. When they're not nesting, they spend most of their time in the marsh, the brackish marshes. That's where they feed, that's where they sleep, that's where they do everything except nest. Okay, so when there are lots of marshes around, you have lots of potential habitat for terrapins. When you don't have lots of marshes around, the opposite is true. Well, as I mentioned, sea level is rising, right? In Maryland, sea level is rising faster than it is in many other parts of the world. Okay, we're in a bad situation here in the mid-Atlantic for several reasons. Not only is the sea level, I mean, we all know sea level is coming up because the ice is melting and the water is getting warmer, but there's something else that's going on in the mid-Atlantic here that's making it worse, and that is that we're sinking. Okay, Maryland is sinking. And so Maryland is sinking, the sea level is coming up. Overall, the net effect is a big effect. The reason we're sinking is because it's a geological response to the last ice age. The ice sheets came down, they sat north of Maryland, Pennsylvania, areas like that. Tremendous amount of weight on the crust of the earth actually pushed across the earth down up there. Well, we were sitting below that. When that part went down, our part came up. Okay, so we ballooned up a little bit. And now the ice is gone and we're starting to sink. So we've got double jeopardy going on here, where we've got not only the water levels coming up, but the land itself is sinking. There's been a lot of work done looking at what sea level is going to look like in the future. I won't go through all the nitty gritty of this. This is taken from a report specifically on Maryland and future sea level. But basically, it looks like from the period from the year 2000 to the year 2050, where sea level will be up between, say, 10 and 19 inches. Okay, by the time the turn of the next century, we're probably looking at somewhere in the area of 1 to 4 feet of increase. This is based on Baltimore specifically, but Baltimore is part of the bay. And one little part of the bay doesn't change that much more than others. So let's look at the bay generally in terms of the elevation. What I have highlighted here are, pay attention to the light blue colors, the light green and the dark green. Okay, those are areas of the coast of Maryland coast that are less than 1 meter above sea level. Based on the projections for sea level rise, those areas will be submerged by the end of the century. The yellow areas are probably likely to be flooded. I hope what you'll take home from this is that an awful lot of the coastline of Maryland is not going to be there anymore in the future. Diamondback terrapins need that habitat. A lot of other species need that habitat as well. We did some modeling work. This was led by my colleague Ryan Woodland, looking at how specific habitats that are important to terrapins will change with time. On the left, we're looking at how many of our known nesting sites will be flooded in the future due to sea level rise. And we've done this under different modeling scenarios, either very conservative or more realistic scenarios. But the bottom line is that by the end of the century, regardless of whether our sea level rise is coming up very rapidly or somewhat more slowly, about 80% of the currently used or historically used nesting areas will be underwater. On the right, I show you the projections for the changes in the two habitat types that the terrapins rely on. The sandy areas for nesting on the left, which is highlighted in red, and in the middle of the marsh areas where they spend the rest of their time. Again, the three bars show 3 different modeling scenarios, but the take-home message is the habitats disappear. If the habitats go away, the animals go away. Okay, that's where they live. That's where they do what they do. Well, our response to sea level rise doesn't help either, because what happens when sea level rise? We try to protect our property, right? We don't want our property to wash away, and so we change the shoreline to protect it. So areas that that aren't washed away as the tides come up or as the sea level comes up will be protected by hardened shorelines, which prevent the animals from being able to use the habitats. On the right is a figure from a paper that was published a while back, showing areas of the Maryland shoreline that are likely or certain to be hardened or protected in the future. And what you can see is pretty much all of Maryland that isn't already hardened is going to be hardened as well. And so basically we're removing a lot of habitat. So the overall, in a nutshell, the sea level rise situation is this. The terrapins require 2 distinct habitat types, brackish marshes and sandy nesting areas. Sea level is rising. It's rising rapidly. The end result of that is that the critical habitats that they require are disappearing. So, just to summarize quickly, the terrapins face a challenge in the future. Warming temperatures are almost certain to affect the sex ratios of the population. I did not mention the second point here, but for ectothermic animals, as temperature goes up, they have to eat. Okay, and so the resource requirements will change as well. It's because they're ectothermic. As they warm up, their metabolism goes up. What feeds metabolism is what you eat. Sea level rise, reducing the feeding and overwintering habitats. Martians, it's reducing the nesting habitats. And finally, there's more of us. Okay, there's 18 million people living in the Chesapeake watershed today. 18 million people, each having a footprint, each having an environmental footprint, and each contributing to what's going on. So I'm finished with this, and I want you to think about this a little bit because we do all have a footprint. Okay? Everything we do has some effect on the environment around us. And especially in a culture like ours, where we are master consumers, we have very big footprints. And unless we decide to lessen our footprints, nothing is going to change. Okay? One of the things people ask me is, well, this is a big deal. What can I do? Well, it's not going to change in the short term. What you can do is what we should have been doing 30 years ago, is reducing our use of fossil fuels, behaving in such a way that we're responsible for ourselves in our environment. So that's where I guess I'll finish up because I think I ran a little long tonight. Just like to thank all the folks that were involved in this stuff, folks across the river with the volunteer program. See my collaborators listed here. And we've been supported initially by the Maryland Sea Grant. The JS Volunteer Foundation has come through with a lot of help for us. And a lot of people have made individual donations to CBL. And a lot of that has, when targeted for terrapin research, has gone to support this sort of thing. And that's what I have to say. Thank you.
Thank you, Chris. We're going to do what I promised. We'll take a question from the audience. I'll take a question from the chat box and we'll alternate for a little while. And if you need to leave, please don't hesitate to leave. So, Ron, you want to take the first question?
Yes, thank you. Can you sex hatchlings as they're coming out of the nest?
No, that's a problem. What we're working on now is a biochemical way of determining sex. So there's this sexual dimorphism when they're adults, but when they're babies, they all look the same. The only way to tell them apart is to look inside, to look at their gonads. We don't do that very frequently because we don't like to. In order to look at the gonads, you have to kill them, and we try not to do that. And so what we're working on now is working with Karis Mitchell-Moore here at CBL to apply a hormonal method of identifying sex, basically looking for a hormone called anti-mullerian hormone. This is based on a study that was published by other people a couple of years ago. This is a hormone that is only present in males. And what it does is it causes male development during the sexual differentiation period. And it tends to remain in the body for some time, so it's measurable. So the short answer is no, you can't tell them apart. And the long answer is there are ways that we're trying to tell them apart. And hopefully we'll get there soon.
All right, thank you, Chris. First question from the chat. Is there or has there been any population surveys to track trends in abundance?
On a large scale, no. Okay, if you say how many terrapins are in the bag, I haven't the slightest clue. There are a few small subpopulations here and there that have been monitored for some period of time. The difficulty is extrapolating what happens in one spot to what happens everywhere else. Okay, so what studies have been done have often come to the conclusion that crab pot mortality is important and continued use of these crab pots without TEDs can lead to population declines, but in terms of actually estimating this or estimating the status of the populations today, there's a lot of guesswork involved. And one of the reasons is they're very difficult. I mean, we're talking about animals that live for decades, right? And animals that lay, throughout their lifetime, lay hundreds of eggs. Only probably one or two of them actually survive. And so being able to track those individuals and put numbers to the populations is extremely difficult.
I was wondering why the commercial crab pots weren't required to have the dollar fixed. Seemingly, they would be the biggest problem.
Yeah. All right. I gotta be careful here. My punk rock cynicism is about to flare up here. So the idea behind limiting this rule to the residential or the recreational fishermen is that people who put crab pots out off of their residential properties typically put them in shallow water. Okay, terrapins like shallow water. Commercial crabbers in Maryland aren't allowed to put their traps in shallow water, but that depends on what you call shallow, right? The rule in Maryland is for commercial crab pots, they have to be in water that's 4 feet or deeper. Terrapins don't know when it goes from 3 feet to 4 feet. Terrapins, we see them in all kinds of situations in water depths. So to answer your question, why? My feeling is that there's a lot of political pressure. There's a lot of money in the crab industry. Okay, it's very important locally to economies, and nobody wants to mess with it. All right, and so that's where we are. Again, that's my feeling from a cynical perspective, but I really haven't seen any other explanation.
Thank you, Chris. This question is a theme for this series. Are blue catfish impacting terrapins? We had them impacting crabs and sturgeon. Can we add another one to the list?
My answer to that is that if there are blue catfish around where there are baby terrapins around, the blue catfish will probably. There's no work at all that I know of that points to blue catfish as being an important population regulator on the terrapins. But like I said, terrapins, especially when they're young, they're just little morsels of food. Okay, and anything with a big enough mouth is going to eat them. And so, yeah, blue cats probably will.
Other question.
So, a sciencey question.
Okay.
You suggested that critical temperature for the local terrapins may be different than 29.3 or whatever it was. Any sense of where that is, where that will fall?
I think it's lower. So the evidence for this comes from, so occasionally, so we do a lot of things with terrapins here, and we sometimes hold them for long periods of time and sometimes not. And sometimes when we hold hatchlings for a while, you have a certain number that don't make it. And when they don't, we necropsy, cut them open and we look at them. And so we can see their gonads. We know were they males or females. And in many cases, we also know the incubation records for those animals. And by looking at those temperatures and the fact that so many of them are females at temperatures below 29.3, it tells me that it is probably lower than that, which is actually problematic because that means the situation is probably worse than we're considering right now. I wish I had the actual value. I've been knocking myself out for 3 years to find the actual value. So
You had a week.
yeah, yeah, no problem.
All right. Another one from the chat. Few animals have the ability to change their sex when needed. Is this something seen in turtles, particularly in terrapins?
No, actually. So we don't see any evidence of sex reversal or anything. With the terrapins, once the sex has been determined, it's determined. Okay, so once you're a male, you're a male, once you're.
Other questions? I thought you'd had your hand up, but no, you were scratching your ear, but Adam, I'll come to you.
So in the controlled nests, is there any way to adjust the temperature of those nests to see whether it might impact the balance?
There probably is a way. We haven't explored it because what we've been doing is trying to do that in the lab. Okay, so what we're doing with the lab is we're using our records from the natural nests in order to make our incubators mimic them. So that allows us to do that, only it's not in the field, it's in here. It gives us more control. Basically. But there, I mean, with enough engineering, we could probably come up with a way. But engineering isn't, well, hasn't proved to be the game.
All right, one more from the chat, and I'm trying to get it. It's about Phragmites. You see a lot around Drum Point and the various methods of remediation of physical cutting, control with Roundup. What's the effect of Phragmites and the effects of control on Phragmites on terrapins?
I can answer that just generally in that when Phragmites takes over an area, it basically outcompetes all of the native stuff. Okay, so the native grasses, the native plants that are typically there, and grasses and the plants define the microhabitats that exist. So when that's replaced with Phragmites, what you have is a different habitat than what you had before the Phragmites today. In terms of the control strategies, you know, I don't know. I mean, it's conceivable, depending on how we do it. It's terrapins one way or another, but we haven't looked into that at all.
So in some of these terrapins that you've been able to track, what's the distance that they typically roam during their lifetime?
And that's a
100 yards or a mile or what?
That's a tough question too. So one of the things is, I didn't mean to imply that we've been able to track the animals in space over time because we haven't. But there has been work in other areas looking at their typical distribution, and it really depends on where they live. There are areas, say along the coast of South Carolina, where they will remain within a marsh creek for almost their entire life. So move very little. There are other areas where they live in small patchy marshes and they have to move around from time to time. And so there are numbers in the literature for estimated home range, but they don't really mean anything unless you know exactly what population you're looking at. It varies from place to place, but we're pretty sure that they don't move real far. Okay, for example, there was work done up the Patuxent River, probably 20 kilometers or so from where our nest is. So here at the Name Ware Station, we're at the mouth of the Patuxent. There was work done up north of there for like 20 years, marking the turtles. And that investigator marked his turtles differently than I marked my turtles. And so if his turtles go up down here, we should see them. We very rarely see a turtle that has a mark. We have seen some unusual marks, but we don't know if they're up there or not. But the point is, they don't seem to move around the whole lot.
Last question.
Does caging of the terrapin nest do anything to the predators around?
To what?
Does caging of the terrapin nest do anything to the predators around them? Like they eat the terrapin?
To the predators?
Yeah.
So the caging of the nest excludes the predators. And so that's the idea behind it. I guess ecologically, we are restricting the predators from a certain amount of food. And so, yeah, from that perspective, the predators might be worse off than they would be. A lot of the predators that we have, these foxes and raccoons, are very opportunistic, and they tend to like to live around people and garbage and stuff like that. And so, I don't know that we ever like starve off all the foxes by caging them. And that's sort of that.
All right, with that, I'll call an end to the questions. Before I ask you to join me in thanking Chris one last time, just a reminder that next week we'll be talking about bottlenose dolphins. And the final week we'll be talking about a history of change in the Chesapeake Bay over the last 50 years or so. I want to add my personal thanks to the young gentleman in the corner who's been amazing tonight. So thank you very much. If you stop by and see me, I will make sure I get a little gift for you for being so great. So thank you very much. Chris, thank you very much. And we look forward to seeing everyone next week. Thanks.